Afternoon, everyone. Fantastic to be here. I'm Duncan. I'm a clinical oncologist, and I'm the chief investigator on one of the REFINE studies. And I guess Matteo, in a way, has set the scene and slightly stolen my thunder. But it seems to be fine to use the excuse that as a clinician we can be slightly immune to some of the more statistical niceties. So I'll play the clinician card strongly at this point. So in terms of, and really just to put a bit more sort of depth and flesh on what we're trying to do with REFINE. So those of you who aren't directly involved in cancer treatment or cancer trials, immunotherapy has really taken off in the last 10 years. I think if you went back a decade, attempts to try and get the patient's own immune system to fight cancer had really got nowhere. And it wasn't really until it was appreciated that there is an immune response to cancer in many patients, but that there are molecular mechanisms by which your normal cells evade the immune system or actually damp down the immune system. So have a sort of cloaking device if Star Wars is your generation, or invisibility cloak if it's Harry Potter. And actually, once you know that there is a way of damping down the immune system, these immune checkpoints, so it's a break that is probably evolutionarily there to prevent autoimmunity, then you can interrupt that. And by blocking the immune checkpoints with drugs that are immune checkpoint inhibitors, you then uncover that immune response. And it really has revolutionized outcomes for patients, many patients with advanced cancers. And I think the most striking one has been in melanoma, where prior to immunotherapies, there was really very little treatment options available. But actually, and patients sadly probably only survived on average around 6 months with metastatic melanoma. But now with immunotherapy, about half of the patients get a meaningful response. And that means that even 5 years later, they still seem to be in remission. And that's probably long-term cure. And that generated the Nobel Prize in medicine in 2018. So the point about this is that we've now got an expanding cohort of patients responding to treatment, but at present remain on those drugs indefinitely. So for many years, and these are the first generation of immune checkpoint inhibitors are all monoclonal antibodies. So they're given intravenously, and they're given every 3 to 4 weeks. And the cost of that in the US is at least $10,000 a month per patient. And in the UK, the list price for pembrolizumab is £5,000 a month. So a huge cost to the NHS, but also a big cost to patients in terms of time toxicity, in terms of repeated intravenous infusions, and also in terms of NHS infrastructure. Suddenly all these patients coming in every month for their IV drugs. So we believe, and I'll go on to explain a little bit why, that there's significant scope to optimize these treatments, trying to minimize patient exposure. And clearly, if we can optimize this and finesse it and work out what is the minimum amount of immunotherapy, immune checkpoint inhibitors to maintain response, then that maximizes the global relevance for these treatments. Because at present, the vast majority of the world's population are not able to access these drugs. So as Matteo has already set the scene, a conventional non-inferiority trial just using a 2-arm design would be huge, but also slightly misses the point. And there are a number of examples in oncology where 2-arm studies have given a sort of winner. There's one at the moment in adjuvant treatment for colorectal cancer where 6 months was the standard of care. There was a huge 2-arm trial to show that 3 months was as good, but we don't really know whether 4 months would have been a bit better, whether 2 months would suffice. So in REFINE, we're taking immune checkpoint inhibitors. We don't know who they work in. There's not really predictive biomarkers. And the initial schedule in licensing was based as if they were chemotherapy drugs. So ramp up the drugs at the doses until you get toxicity, and then pick the lower dose. And actually, if you go back to those phase 1 studies, there was very little evidence of a dose-response effect. And there were patients responding to very, very low doses, probably a 20th of what's given clinically. And in those patients that didn't respond to low doses, they didn't respond to dose escalation either. The other point is that the clearance of these antibodies falls as patients' health improves. So the half-lives of the monoclonal antibodies actually lengthen as patients improve. And in silico work done by the NCI in the US shows that already the half-lives of these drugs are probably twice the frequency that we're giving them, and that they maintain what we think is well into the therapeutic range doses for at least double, if not longer, treatment intervals. So that really is the underlying rationale for REFINE. So we talked about ways that we can use MAMS-ROCI designs to test overall shorter durations, but in REFINE, an alternative is to test different extended frequencies using a multi-stage approach. And this is a basket design because actually these drugs are now being used in more and more different cancers. And so you can have tumor-specific cohorts where you ask that question. So there are two REFINE trials, slightly just historically. There's REFINE Lung, run from Imperial, which I'll talk about in a minute. And at the MRC CTU, we're running REFINE. And REFINE Lung was originally called something else. So we came up with the name REFINE, or actually I think it was Ruth. And REFINE Lung thought that was a lot better, so changed them from Convolute, which it was going to be, to REFINE Lung. Our REFINE takes an initial two-arm trial to show feasibility, and as Matteo said, in a conservative way, because what we're trying to do is optimize treatment. We're de-escalating, which has historically lots of challenges with sort of clinicians and patients, though patient buy-in has been excellent so far. Initially demonstrating that you can randomize patients to an extended interval, which is twice the standard of care, to exclude a major loss of efficacy and show the feasibility of doing this, and then progress, as Matteo alluded to before, to a 5-arm trial. And the idea of the sort of MAMS-ROCI approach is that across the x-axis we've got the interval that's extending, and then we've got a hazard ratio for progression-free survival versus standard of care to find the sort of the acceptable range. And as Matteo said, in this instance, 12 weekly treatment would be our optimal treatment choice. So REFINE currently has a renal and melanoma cohorts open in 12 centres. Recruitment has been really challenging. We got the grant for this. It's funded by the John Moulton Charity Trust. We got the grant for this just before COVID, which gave us a little bit of a buffer of time to work up the study. But those of you who are working in NHS sites at present just know the huge challenges that we face. REFINE Lung, run through Imperial, is funded by the NIHR. And the slight difference here, so in REFINE Renal and Melanoma, we're taking patients who've had purely immunotherapy and who are responding at 3 months, and then randomise them into the extended frequency versus standard of care. In lung cancer, standard of care for patients is that they get chemotherapy and immunotherapy in combination, and so they're randomised, those patients who are responding, at the end of the chemotherapy component, so that's at the 6-month mark. So there are some subtle differences between the two approaches, which is another reason why it's not a sort of single basket. But both trial teams are working closely. And actually, these the two trials, REFINE and REFINE Lung, sit as part of a whole range now, actually, of global optimizing immunotherapy trials. And it's interesting here how people are taking different approaches. So there's the early cessation work. Matteo mentioned, can you stop in patients who are responding? Then there's a cohort of extended interval ones, of which ours sit. And then there are some studies, particularly in India, testing, can we just lower the dose completely? And so it'll be fascinating to see how all of this pans out. And there'll be a lot of overlap, and there'll be a lot to learn. This is particularly in the world of immunotherapy, but across cancer, I think now we're looking at a whole range of other approaches and other uses of MAMS-ROCI in this arena. Slightly keeping to time. So yes, with the specific question of immune checkpoint inhibitors, there's a real opportunity here to optimize those, and there's potentially huge benefits all round. I think this is a fantastic exemplar for, as I say, future cancer treatment optimization trials. It is very much the domain of academic trialists. These trials actually save a fortune. in terms of drug costs. And in fact, there are some discussions as to whether you could make these trials pay for themselves. And the Dutch government and health system has started doing that. The basket approaches and novel methodologies are absolutely vital that we can be as efficient as possible and answer these questions quickly. And as I mentioned, there remains huge daily challenges for all of us trying to do academic work in the NHS. And on that note, thank you hugely to the Refine trial team. Matt Nankerville's here with us today, and everyone at the CTU who supported this. Obviously the funders in terms of John Moulton. Big credits to the Refine Lung, TMG, and particularly Michael Sackle at Imperial and Ehsan Gorani. And then also support from the pharmacology people at the NCI, Cody Peer and Doug Figg, who are going to do our pharmacokinetic work and really underpin the scientific questions in this. And then support from colleagues at the Optimal Cancer Care Alliance, who are really trying to promote these kind of studies worldwide. So that's all I was going to say, and obviously there'll be time for questions.